This is our skeletal muscle cell or muscle fiber. We talked about the components of the muscle fiber that allow it to function the way it does. But now we're going to zoom in even further and examine the sarcomere, which is the structural and functional unit of the skeletal muscle cell, in that it is the smallest structure that is capable of contraction or shortening. This means that the movements that the entire muscle organ engages in are due to the movements of these microscopic sarcomeres. It's pretty amazing when you think about it. The muscle fiber's main ingredient is the myofibril. We talked about that. These bundles of very thin thread-like proteins called myofilaments. Remember, each myofibril is the bundle like a sewing thread. It's composed of much thinner myofilaments, like the thin threads that compose a sewing thread. And it's these myofilaments that we're about to examine. You can see here they come in two flavors. We have actin, the thin filament, myosin, the thick filament. The thin filament is structurally more complicated in that there's three main proteins that compose it. The major structural protein here is actin. Actin is going to resemble two strands of beads or pearls, kind of like Mardi Gras beads. Each protein contains a myosin binding site that myosin is capable of sticking to. Attached to these actin balls are two regulatory proteins, troponin and tropomyosin. Troponin contains an attachment site for calcium ions. When calcium ions are present, they will bind to this attachment site and change the shape or configuration of troponin. So it has one shape when there's no calcium attached to it, and a different shape when calcium is there. Troponin is attached to tropomyosin, which is a thinner, more thread-like protein, which sits in the grooves of actin. So as the actin strands twist together, they form a groove, and tropomyosin will be found there. Its location is right on top of those myosin binding sites. So it will sit on top of them and block them when the muscle is at rest. Let's take a look. So we're looking at close up here of the myofibril, right? Actin here. And when we zoom in, we see that it's a bunch of these little purple balls or beads or pearls. And these are our actin proteins arranged in two strands. Each protein has a spot on it where it can stick to myosin. Troponin here, there's no calcium on it right now. So this is one shape. When calcium attaches to it, it will change shape. It is associated with tropomyosin. It sits in the groove here. So when troponin is bound to calcium and changes shape, that shape change moves tropomyosin. It moves it specifically so it uncovers these myosin binding or attachment sites. So this would be troponin when the muscle cell is not contracting and at rest. No calcium is present, the cell is not being stimulated, so it's not contracting. Myosin is more straightforward. It makes up the entire thick myofilament, and it looks like a bundle of golf clubs with the heads pointing out and the handles all grouped together. So here, myosin, or the thick myofilament, it's named the thick filament because it's simply thicker, more protein overall. And close up, it looks like this, with it's called the myosin tail or rod, grouped together or bundled together. The myosin heads all point out. Myosin is special because it is capable of breaking down ATP chemical energy. It has within that head an enzyme called ATPase. Whenever you see a something ACE, that's an enzyme. And fortunately for us, they're usually named for what they do. ATPase hydrolyzes or breaks down ATP to access the energy that's contained in it. So myosin is capable of breaking down ATP to convert chemical energy to mechanical energy. When this occurs, myosin uses that energy to move. It's able to shift the head using that chemical energy. And finally, we've already hinted at this, but myosin is capable of binding to actin at that binding or attachment site. This means then that when myosin and actin are attached, myosin can move, and when myosin moves, actin will move also. A common way to illustrate this is through a love story. Actin and myosin, kind of like Romeo and Juliet, star-crossed lovers, forbidden lovers. Myosin's a bad boy. Actin is a beautiful, pampered girl, only child, very overprotective parents who are constantly monitoring her activities, her friends, and her social media. But they love football. Whenever the Saints game is on, they make a bunch of food, snacks, beer, have their friends over, and for a couple blessed hours, they're not breathing down Actin's neck. These parents are like troponin and tropomyosin, right? They're different proteins, but like parents, they co-parent, they cooperate to regulate what actin is doing. So we got our overprotective parents, regulatory proteins, troponin and tropomyosin, stuck on our beautiful actin. Now, calcium is an important player here too. Calcium is like that Saints game. Whenever it's around, it's distracting. Troponin and tropomyosin, causing that shape change to occur that moves tropomyosin out of the way. So it's kind of like actin can have her phone for a minute, and do whatever she wants on it unmonitored. What's she going to do? Well, she's going to contact bad boy myosin, who crawls through the window, and they can become now attached. And myosin is going to take her away. And when he moves, she moves. And the muscle is capable of movement. Right, so again, troponin, tropomyosin are regulatory proteins or parents. In the presence of calcium, distraction, Saints game. These guys shift out of the way. They're momentarily distracted. Attachment site is uncovered, and myosin can stick to it and take it away. These thin and thick filaments are organized in a repeated pattern within the skeletal muscle fiber. This creates those lovely sarcomeres, which are so pretty, but they're also integral to how the muscle functions. So we have to examine in even more detail. Again, this is the structural and functional unit of skeletal muscle cells. It's very important. This is the smallest unit that's capable of contraction. So, when the sarcomere shortens, the whole muscle fiber shortens. When all the muscle fibers shorten, the fascicle shortens. And when the fascicles all shorten, that whole muscle organ shortens and pulls on the bones that it's attached to, allowing us to move throughout the day. The sarcomere stretches from Z-disc to Z-disc. The Z-disc is a plate or disc of proteins that forms an anchoring point for actin of the thin filament. So all of our actin thin filaments will be attached here and point towards the middle of the sarcomere. In the middle, we have the M line, which is an attachment point for myosin of the thick myofilament. So think M for middle, M also for myosin of the thick filament. So here we have two sarcomeres, 1, 2. The Z disc here, here, and here. You'll see that it is zigzagged in the diagram, and it's actually zigzagged in real life too, though this is a bit too low of magnification to see that. It just looks like a squiggly line, but you can almost tell. Attached to the Z-disc are the actin thin filaments. So we have a Z-disc here, thin filaments attached pointing in, Z-disc here, thin filaments attached pointing in, and then the next sarcomere. Same arrangement. Middle is the M line, which forms an attachment for myosin of the thick filament, pointing out, opposite direction, towards each Z-disc. So, again, M for middle, M for myosin. Maybe think Z for the ends. Z is at the end of the alphabet, Z-disc is at the end of the sarcomere. We can see here, because of the way these filaments are attached, there's areas where there's thin filaments, there's thick filaments, and also overlap between the two. Of course, these have names. The I band is the area where there's only thin filaments, only actin. This spans each Z-disc. So each I band is going to go into 2 sarcomeres, a little bit into one for one side of the Z-disc, and a little bit to the other side of the Z-disc into the next sarcomere. This is the part of the striations that appears light because we're talking thin filament, less protein. This gives us the light band, light stripe of our striations. Versus the A band. This spans the entire length of myosin, or the thick filament. Myosin is going to make a thicker filament with more protein, so this A band area stains darker. This is our dark stripe or dark band. Within the A band, there's an even darker area where there's overlap between the two. And it's this overlap where actin and myosin can meet, where thin and thick filament touch, and the thick filament can grab the thin filament. That allows contraction to occur. This last area is the H zone. It's right around the M line where there's no overlap and only myosin is found. It's a bit hard to keep straight, so let's take a look at it. I band, only thin filament. So, little bit in this sarcomere, little bit in this, spanning the Z-disc. A band is where there's myosin. So it spans the M line, because that's right in the middle of the myosin, goes towards either direction, and encompasses 2 parts. This area where there's overlap between thick and thin, myosin and actin, and also this H zone where there's no overlap and just myosin. Maybe you can think of that as the end zone, H zone, end zone, before you get to the M line, perhaps. And then the words light and dark can kind of help you keep these straight. The word light has an I in it, so light, I band, there's only thin filament. The word dark has A in it, so dark, A band, where there's thick myosin and also that zone of overlap. So in this way then, myosin can grab actin and pull actin, the thin filament, towards the middle. So these myosin heads are grabbing actin all along here, grabbing it and pulling, pulling, pulling. And then same thing over here. Myosin grabs actin, pulls it towards the M line. So we'll start to see the I band shrink, right? And the H zone shrink, as these are the areas with no overlap. And this zone of overlap is going to increase as the sarcomere shortens, right? So Z discs will move closer together. More of that actin is going to overlap with the myosin until we'll see in a fully contracted muscle there's pretty much no H zone, totally disappears as we see more overlap here. And there's almost no I band either, right? As this area is going to also see overlap. So you can kind of imagine myosin grabbing actin here and here, and just two arms grabbing two ropes in opposite directions, and then you pull them towards yourself. Right? Pulling the Z discs closer together and shortening that sarcomere.